Hello.
Hello?
Hi, Mark. How are you?
Good, good.
Uh, okay, so today I thought we'd go through some paper ones. How does that sound? Just pick up where we left off.
Okay.
Um, and if you want to go through a paper two instead, I'm happy to. I thought
Yeah.
let's
Paper ones are good. Paper ones are good. Yeah.
okay.
Start
Perfect.
paper ones, so that's good.
Cool. So this is the paper we were doing last week. We'll just pick up where we left off.
Okay.
Start with question 15 then. Can you just walk me through it? What's the answer and why the other one's wrong?
Okay. Okay. Um, so first of all, it's not exocytosis, it's endocytosis. So B and D are wrong. And then it would be binding to a protein receptor. So it's C, not A.
Good.
And yeah, because protein receptors are, yeah, they allow for endocytosis.
Yeah, which part of the virus is going to attach to that protein receptor?
Um, like a specific part of the virus that has complementary shape.
Yeah, so what are the different parts of a virus?
So um, there's the genetic information surrounded by a protein capsule.
Yeah, good. Do you know what that protein capsule is called?
No.
It's capsid. Does that ring?
Capsid. Okay.
Yeah, so you've got the capsid that's made up of capsomeres, which are just subunit protein subunits.
Yeah.
So that protein coat, the capsid, will bind to a protein receptor, cell surface, and then endocytosis and so forth. Okay, good. Next question.
Mm. Okay. B.
Okay, talk me through that.
Okay. The so by the uh so when the fatty acids or phospholipids are unsaturated, the hydrocarbon tail bends.
Yep, good.
So there's more space for the phospholipids to like move around, so it's more fluid.
Yeah, good, perfect. And in terms of the effects of temperature on the fluidity, what's the actual effect of changing the temperature on the fluidity?
Um, so like cholesterol uh regulates the fluidity. So when there's an increase of temperature, the cholesterol makes sure that it doesn't get too fluid, or when there's a decrease, then make sure that it's fluid enough.
Yeah, good. Okay. Next question.
Okay. B.
Good. And what do they mean by inversely proportional?
That means that so if you put on the graph 1 over uh distance squared, then you would have, and if the other axis is rate of diffusion, it would be a straight like linear line.
Yeah, good.
So
So
as distance increases, rate of diffusion increases, and as the size of whatever's diffusing increases, the rate decreases.
yeah, good, perfect. Next question.
I think this is like meiosis,
Yeah, that's right.
and I like they might have done it in 2009, but I'm pretty sure we don't do it now.
Okay, all right, that's no problem. The answer is B, because
Okay.
if you imagine if you had mitosis in your gamete, then when the two gametes fuse, you'd have double the chromosome complement.
Mm-hmm.
It's not what you want. In a zygote, you just want um, however many chromosomes the species normally has in their cells. So the answer is B, but that's fine. We'll move on. Um, question 19.
Uh, so it'll reach the metaphase.
Yeah, good.
Yeah. Because in the anaphase, that's when the chromatids are separated.
Good. That's right. Okay, next question.
Oh, okay. CIU.
Yeah,
Yeah.
good. Explain that to me.
So so the triplet code is CIT,
Yeah.
which means that the mRNA codon would be GUA,
Yep.
and then uh, by complementary base pairing, the anticodon will be CAU. So like the anticodon is like the same as the DNA
Yeah.
uh, triplet code, but instead of thymine, it has uracil.
Good. Perfect. Next question.
D.
Good. Now explain to me the concept of semi-conservative.
Okay, semi-conservative is when the DNR is replicated by using both strands of the DNA as templates
Yeah.
and creating two molecules with both an old strand and a new strand out of
Good.
one DNA molecule.
Good. And those old and new strands are sometimes called parent and daughter strands. Um, in terms of translation, so the question says it occurs at the surface of the ribosomes. Where are ribosomes found in cells?
On the rough endoplasmic reticulum,
Yep. Anywhere else?
in the cytoplasm.
Good. Yeah. So they can be free in the cytoplasm or on the ER.
Okay.
Okay, next question. I'll try and scroll so you can see the question and the answer. But let me just know when you finish reading.
Okay. Oh, this one looks a bit. Okay. Um. Okay, so the percentage of cells containing N15 decreases
Good.
as the generations go. So um, it wouldn't be A or wouldn't be B.
Okay, good. Now, is it C or D and why?
Yeah. Um, I think it's C.
Okay, talk me through it. That's right, but talk me through it.
It's because the nitrogen 15 will never be uh, 0, like percent. So we just talked about semi-conservative replication.
Mm-hmm.
So um, the okay, when you look at one cell and like one chromosome of the cell, then in the first generation, the so one DNA strand will consist of both a strand of a template which is 15 and
Yeah.
then another template which is 14, and that twice, right? But
So both of the templates will be N15, but the growing strand will be N14.
yeah, so you'll just get like two sets of N15, N14.
Yeah, good. So you're
Yeah.
essentially halving
Yeah.
the cells containing N15 with each generation.
So it'll be like 50%, and then it'll go down to 25%.
Good. And
Um,
you said at the
it's always half.
sorry?
It'll half as generations. Like the percentage of cells containing N15 will decrease by half the value that it was.
Yeah, good. That's correct. At the start of your answer, you said that it will never reach zero.
Yeah.
If we were looking at like a sort of mathematical equation where you're always halving the product, then that would be true. But because we're talking about nitrogen atoms, at one point you're going to have a cell that only has one N15,
Mm-hmm.
and then to replicate the DNA, it's going to have to, it will pass it on and on and on. And what you might have is one N15 remaining in the cell in like one strand of DNA.
Yeah.
Because of mutations and repair that will occur just naturally
Oh,
after
okay.
after so many generations. Like now we're talking, about thousands and thousands of generations
Yeah.
to lose that N15. Um,
Okay.
but yeah, that was just an extra point. But good, that's quite a tricky one. Let's move on.
Yeah. Okay. Yeah. It's A.
Good. So what does RNA have, and what does DNA have, and what are the differences?
So RNA has uracil, uh, and DNA has thymine,
Good.
and RNA has ribose, and DNA has deoxyribose.
Good. Perfect. Right. Next question.
Hmm. Okay, so it's definitely not A and B because it's loss of water vapor from leaves,
Good.
transpiration. D.
Yeah. Good. And why isn't it C?
Because it doesn't maintain. Well, I know that it's D because if the stomata are open, CO2 can diffuse into the plant for photosynthesis
Good.
as a carbon source. It's probably not It's not maintaining the water potential because it sets a gradient for water to travel up the plant into like from the root to the leaves.
Yeah, so it actually causes the plant to lose
Yeah, it
water.
doesn't maintain. Yeah.
Yeah. Okay. Good. Next question.
B?
No, it's
Kind of
not.
a guess.
It's not B. But talk me through your reasoning, man.
um, not a lot of reasoning actually. Okay, wait. Okay. So A is not correct.
Good. Why isn't A correct?
Because the mineral uptake of root hair cells, it doesn't decrease because root pressure is decreases. It has nothing to do with that.
Good.
Yeah.
Good.
And then okay, for C, well, translocation could decrease at night because less assimilates are, well, no assimilates are being formed because no photosynthesis occurs at night because the stomata are closed.
That's true, but you still do have assimilates in the source cells stored in the vacuoles.
Yeah.
Throughout the night, they will continue to diffuse into the and move into the.
Yeah, because it might go like to storage or something. Oh.
Yeah, exactly. The plant pretty much needs a constant movement of assimilate. The rate could decrease at night by a small amount, but it's not, the rate isn't linked necessarily to photosynthesis because the source cells themselves have vacuoles that they can store assimilates in during the day so that at nighttime they can continue to release them.
D makes sense. Yeah, D does make sense because so when there's transpiration, it's like when you have a straw, um, you create negative pressure.
Yeah.
So the stomata are closed at night, so no transpiration occurs at night. So, yeah.
Yeah, good. So at nighttime, you close the stomata, you've got no transpiration, you don't have that negative pressure, so there's less tension and the vessels expand.
That makes sense. Yeah.
And why is B incorrect?
Um, so there wouldn't be buildup of water within the stem. Like, if anything, there might be, well, there wouldn't be intake of water, I think, or maybe it would be.
Yeah, exactly. So you've got an equilibrium with water coming in at the root and leaving at the leaf.
Yeah,
And you just close the exit.
nothing occurs then. Yeah,
Exactly. You'll get no movement.
yeah.
So good. Let's move on.
D.
Good. And why isn't it C?
Because it can't travel in two directions in one tube.
Yeah.
At the same time.
Okay. Question 27.
I would say 1 and 3, so B.
Yeah, that's correct. Why is 2 incorrect?
Because sodium ions are actively transported together with well, actually they're not. Then okay, so I know that sodium ions together with potassium ions can be transported by active transport.
Good.
So that's why I said no.
Okay, in terms...
But I think that's something to do with the nervous system, maybe. I don't know.
So the key point here is that sodium and other ions like potassium can go through the membrane, but they can't diffuse. They can't freely diffuse through the membrane.
Oh, so facilitated diffusion. Okay.
Good, exactly. So you're right. They can be actively transported against the gradient if the cell has the machinery to do so. But in many cases, diffusion is still occurring, only it's facilitated diffusion through channels or carriers.
Okay.
And the reason that is because sodium has a charge, and therefore, although it is a small atom, it's going to be repelled by the phospholipid bilayer.
Yeah.
Whilst oxygen and other inert gases don't have charge, they just freely diffuse.
Yeah.
Okay. Next question.
I would say tertiary and quaternary.
Good, that's correct. Talk me through it.
So, so the 3D structure of the 3D shape of the hemoglobin changes, which is tertiary structure.
Good.
And then quaternary structure is so it's the interaction between the different protein chains, or polypeptides.
Yeah.
But it's also the interaction between the heme group and the polypeptides.
Yeah, good. So as the 3D or tertiary structure changes, that causes downstream changes in quaternary structure.
Yeah.
That's what allows the bonding of oxygen to happen more readily.
Yeah.
Okay, let's do this question and then take a break.
Okay. C.
Good. Talk me through it.
So okay, so at at the partial pressures of oxygen, the vertebrate is has less of a saturation, so the hemoglobin are less saturated. So they will release oxygen more readily because the hemoglobin has a lower affinity to
Yeah. So hemoglobin. So if we said vertebrate X is exercising and the oxygen demand increases in its muscles, it would have less oxygen in the hemoglobin
yeah,
that's reaching its muscles, and therefore, because of the binding properties of oxygen to hemoglobin, it would be released more readily.
yeah.
Okay, good. Let's take a 5 minute break and pick up at half.
Okay. Hello.
Hi. Okay. Let's get started then. Question 30. Hello?
Sorry. I'm just reading.
Sorry, that's okay.
It's not B or um. It's not wait. Yeah, it's not B. It's not D.
Good. So it's either carbon
It's
monoxide.
either carbon monoxide or nicotine. Um, I think it's nicotine.
Yeah, good. So how did you come to nicotine then?
Okay, so I know that carbon monoxide, so what it does to the cardiovascular system is, or circulation is, it creates carboxyhemoglobin, and I know that it damages the arterial lining and forms plaques, can lead to plaques being formed.
Yeah, good.
Um, and I know that nicotine increases blood pressure, um, and it also damages the lining of the arteries and can lead to buildup of fat,
Yeah,
which leads to like macrophages coming and engulfing it. But um, I think then something to do with platelets. So I knew by knowing that carbon monoxide doesn't do it,
yeah,
and
good.
carcinogens don't, and tar doesn't. So nicotine must do it. I don't know.
Yeah, good. And I mean, you're probably going to have to use exclusion for a lot of the trickier questions. But yeah, if you were thinking about it in the sense of like nicotine is a complex molecule that will bind to cell surface membrane, therefore platelet cell surfaces, whilst carbon monoxide is again a gas which is more likely to diffuse into cells and have effect on their, sort of cellular machinery. And I
Yeah.
mean, theoretically, that could make a blood platelet more sticky. But what's a bit more intuitive, I guess, is that nicotine by binding to cell receptors could cause platelets to clump together if it had more than one binding site.
Yeah.
But yeah,
Definitely.
good.
Makes-
Fine.
Yeah.
Go on to the next question.
Mm, one, two. Okay, so I know that two's definitely right. Because it says extra mucus is produced. Oh no, exocytosis. Okay, sorry. Two is incorrect. So um B.
Yeah, good. And why? So talk me through one and three then.
So one, because the ciliated epithelium activity increases because the cilia has to beat the mucus up towards the larynx to be swallowed.
Yeah, good. And
Um, sorry.
how about number three?
Yeah. Three. I don't really know why. My best guess maybe would be the. I don't know. I actually don't know.
Okay, so what does faster respiration correlate to? And if any in any cell, if respiration is increasing, what does that correlate to?
More ATP, more activity.
Yeah, good. So more ATP, so the cell is basically using energy. So you think about it in the sense that an increase in respiration directly correlates to an increase in activity.
Which leads to increase of uh, contraction or relaxing. I don't know.
Okay, so all muscle cells can only do two things. They can either contract or relax.
Yeah,
One of those is an energy intensive.
contracting is energy, relaxing not.
Yeah, good. And so in the question stem, it says the tubes of the gas exchange system narrow.
Mm-hmm.
That's caused by smooth muscle contraction. And so it would make sense then that the smooth muscle cells are respiring faster. Does that make sense?
Yeah.
Okay, good. Let's move on. Question 32.
So B lymphocytes more active
Why?
because if pathogens enter the airways, then it means that an immune response could be triggered.
Good, yeah, and why is that? So we're not talking about pathogens. We're just talking about minute particles from a very dusty environment.
Well, even well then hmm, macrophages would, I guess, engulf the those particles, and it could lead to inflammation,
Yeah.
maybe, um, which
So
could
what's what's the key step that activates the B lymphocyte?
activate.
Yeah,
So it's either helper T cells being activated and then secreting cytokines,
good.
or it's the B cells themselves binding to the antigen.
Good. So, and I mean, in the case of the helper T cells, they'd also be binding to an antigen, right?
Yeah.
The activation of B lymphocyte kind of requires binding to an antigen. And the environment we're talking about here is just a person inhaling minute particles. They haven't told you that there are pathogens. They haven't told you anything about the particles other than the fact that they're small.
Mm-hmm.
And so you can say with sort of a reasonable educated guess would be that those particles are going to get broken down by macrophages in the lung.
Mm-hmm.
Somewhere in those particles, there is going to be some protein. And the idea is no matter what the protein comes from, if digested by a macrophage and presented to T cells or B cells,
Mm-hmm.
that's going to make the B cells more active.
Yeah.
Good. And how about goblet cells then? Would they become less active or more active?
Um, they become more active.
Okay, good. Why is that?
Because more mucus will be produced because it like traps uh the particles. So when the particles enter the airways, they're trapped by the mucus.
Yeah, good.
And then that mucus is then beaten up the airway by the cilia and needs to be replaced.
Good.
So goblet cells are more active.
Good. Yeah, good. And that's exactly what the mechanism is. It's not the fact that there are more um, dust particles or whatever in the in the bronchioles and the bronchus. It's more the fact that because they're getting trapped and the mucus is getting removed, there's then a lack of mucus, so the goblet cells will increase their activity to sort of maintain that equilibrium, and that will cause in total a greater amount of mucus. Okay, good. Let's move on. Question 33.
Oh, okay. Wait. Natural. Natural. Artificial. Okay. It's B.
Okay, talk me through it.
So active is when one produces the antibodies themselves. Passive is when the antibodies are foreign, and enter the body. So natural is when an infection occurs and causes an immune response, like naturally, and then artificial when injected, so vaccinations.
Yep.
Artificial active. Natural passive is when, the antibodies are passed on naturally from mom to child, but the antibodies are still foreign.
Yep.
And then artificial passive is when antibodies are injected, maybe because you have an, I don't know, acute disease or something like that.
Yeah, good, exactly. So injected antibodies are often used to treat conditions like rheumatoid arthritis, where you have an autoimmune reaction.
Yeah.
You can just inject antibodies against, for instance, B cell antigen, and that will reduce the response. So artificial passive is often used in controlling diseases where you don't want to destroy all your B cells, for instance, but you might want to just limit their activity. But yeah, all the rest are correct.
Okay.
Question 34.
Uh, okay, it's not C because T cells don't produce antibodies.
Good.
Um,
You can eliminate another one of the answers with that same logic.
oh, yeah, D.
Good. So you're left with A and
A
D.
and D. Um, yeah, it's B.
Um,
Or not.
so
Okay.
what type of cell makes and releases antibodies?
B cells do. Well, plasma cells.
Yeah, okay. So that's the distinction they're testing in this question. So can you do
Okay.
plasma cells that make and release antibodies as opposed to just undifferentiated B cells? It is like a seemingly unfair question, but actually A and B are pretty accurate. The only one that has a tiny bit of incorrectness, as it were, is B. So the single best answer is A.
Okay. No. Yeah.
Let's move on. I mean, the thing with these questions is that often they'll have one or two that are just obviously wrong
Yeah.
material, and then one of them is going to be a distractor. And in 34, the question answer B is definitely a distractor, and they'll put in a few questions like this in each paper to try and like limit the amount of people who get 100% in the exam.
Yeah.
So I really wouldn't worry about questions like this. They make up like such a minute amount of the questions, and you can still easily get an A with getting like two questions like this wrong.
Totally.
Let's move on.
Okay.
So question 35.
C.
Good. So talk me through each of these answers.
Okay, so first one, they become active only when a specific antibody binds to the surface receptor. It's not true for so it doesn't only become, so it could be that a specific antibody, like a foreign antibody, binds to one of the surface receptors and they activate, but it doesn't have to be.
Yeah, good. And I mean, if they change the word antibody to antigen,
Then
that's
yeah,
good.
then that would be correct.
Yeah. Okay. So how about B?
Um, so B cells don't actually meet an antitoxin in a cell.
Yeah.
Yeah, it's T killer cells too.
Yeah.
They don't. They're not even like activated when that happens. They're actually activated when helper T cells produce cytokines.
Yeah. Good. Let's see.
And then D. Um, that's T cell helper T cells.
Yeah.
Yeah.
Good. All right. Let's move on. 36.
Hmm. Okay. Hmm. It's not D.
Why?
Because like the purpose is not to kill resistant strains of bacteria.
Yeah. I mean, you do want to kill resistant strains, but it's not specifically for that.
Yeah.
Yeah. Good.
Um.
So I guess when you're
Sorry.
sorry, go on. Give me
Um.
thoughts.
Okay. I think it's to maintain the concentration of oh no, it's antibiotic. Okay, wait. Uh, it's C.
Right. So in this question, C is the distractor.
Ah,
You were right with B.
is it okay?
So C is correct in the sense that you want to tell the person to finish their antibiotic treatment to prevent the development. But preventing the development of resistant strains doesn't really have anything to do with taking them at evenly spaced time intervals.
Okay, I just thought, yeah, makes sense. It could be for a longer time, could be for a shorter time, evenly time. Yeah.
Yeah. And I mean, also with this question, you can think about it in the sense that, you know, the reason why we design methods to give people antibiotics in a certain way, the primary reason is never going to be necessarily to do with resistance. It's always to just get rid of the bacterial infection, as it were. Our primary concern is treating the patient, and then the secondary concern is like the sort of public health problem of dealing with resistant strains of bacteria. So primarily, you want to keep that concentration over the minimum therapeutic dose, um, and secondarily, you want to finish the course so that you don't have the development of resistant strains. And A is definitely wrong because
Yeah.
you want a lethal dose immediately, because if you slowly introduce the bacteria to antibiotic, then the bacterial colony will slowly start selecting for strains that are resistant.
Yeah.
Okay, good. Question 37.
D.
Good. Not going to labor those points.
Yep.
38.
Um. This is not part of our syllabus.
No worries.
So isn't this? This is also like ecosystem. Yeah.
That's the end of this paper then. Good. You happy to go through another paper one?
Yeah, that's fine.
Okay, I'll get one up now. All right, we'll do 2008. All right, start with question one.
Okay. C.
No.
Oh. Well, okay. So lysosomes are the smallest.
Are they?
Oh, the nucleoli? I don't know.
So the smallest is actually the centriole.
Oh.
So that would instantly give away the answer if you thought about it in that sense. Centriole is like it's like a minuscule organelle. It's not even an organelle. The centrosome is the organelle, and the centrioles make up this centrosome.
Hmm.
And you have two of them sitting at right angles to each other, sort of like cylinders, which are microtubule organizing centers. Um, Lysosomes are going to be much bigger than centrioles because they're a fully functioning organelle that has to. If you think about lysosomes in the sense that they have to sometimes engulf bacteria when they fuse with a phagosome, that kind of thing. It sort of makes sense why they might have to be bigger. Nucleoli here is a bit of a distractor because I think the the sort of jerk reaction is to assume that they're talking about the nucleus, which is a huge organelle, but actually the nucleoli is smaller than the mitochondrion. But yeah, it's a bit of a brutal first question for that paper, but okay, let's move on. Question 2.
Oh, okay, wait. Um, one. Four. 1423. Yeah. A.
Good. That's correct.
Yeah.
Right.
Oh.
We,
Am I allowed a ruler?
yeah. You have a ruler in the exam, but we'll just skip it for now because I'm pretty happy that you know how to do these questions.
Yeah.
Um, yeah.
What's the expression? So it's not 70S ribosome.
Good.
It's not plasmid.
Yeah.
Um, chromatin is DNA plus histone, which I think is present in both. So I go for mesosome.
So it's actually chromatin,
Oh,
not
I
present.
don't really know what mesosome is.
That's okay. I mean, it's not in the curricula at the moment, but a mesosome is sort of a folded invagination in the plasma membrane.
I yeah, I guess that uh, bacteria having a circular DNA, they don't have the
Histone.
nucleosomes really.
Yeah. Good.
Yeah. Okay.
Um,
Makes sense.
don't worry about mesosome. It's like mesosomes. They're actually just an artifact of preparing bacterial cells for imaging. They don't have any function. It's just if you if you chemically fix a bacterial cell and look at it under a microscope, you see invaginations of the plasma membrane, and that's it isn't a functional thing, so you don't have to worry about it.
Mm.
Okay, let's move on to question 5.
Oh. It's definitely D
And why is that?
because hydrolytic enzymes are present in lysosomes. So
Yeah,
yeah.
yeah, perfect. And you don't need to worry about mitochondria versus nucleus.
Yeah. Okay. Wait. Using Benedict's blue. Okay, so it doesn't contain X, which is a monosaccharide. So it would have to be B.
Yeah, sure.
Yeah.
Yeah.
And I guess Y is a non-reducing sugar.
Yeah.
About disaccharides, there's one disaccharide that is reducing sugar, isn't there? Or like one well-known one, maybe?
So there is a group of disaccharides that are reducing sugars.
Mm-hmm.
Um, so you've got like lactose and
Like,
maltose.
yeah.
But yeah, I wouldn't really worry about that. I don't think that they really test that fact in terms of looking at chemical structures anymore. This is like a really old paper, so I wouldn't really
Yeah.
worry.
Okay.
Question 9. So question 7.
Hydrogen bonds.
Yeah, good. Question 8.
B.
Okay, good. Why?
So cellulose, uh, so they would be flipped by 180 degrees.
Yep.
And then amylose do have alpha glucose 1 to 4 linkages.
Good. Question 9.
D.
Yep, that's right. And why is that?
So they are not soluble in water, so they're not attracted to water because of their large, uh, their fatty acids. So they're large non-polar uh, hydrocarbon
Good.
fuels.
Right. Question 10.
pH change.
Yeah, good. And why isn't it the other answers?
So condensation and hydrolysis have to do with peptide bonds,
Good.
while ionic bond is between R groups.
Good.
And then hydrogen ions can interfere with ionic bonds, but temperature not.
Yeah, good. I mean, temperature.
Well, it could maybe, but I think like it's just more. It makes more sense.
Yeah, it does make more sense. Also, they're talking about breaking ionic bonds specifically, and if you decrease temperature, you aren't going to be breaking any bonds because
Oh
you're
yeah.
increasing the kinetic energy of the system. Does that make sense?
Yeah, but then how does temperature alter the? Because there's like optimum temperature, right?
Yeah.
So, uh, so you'll have like a peak, won't you?
Yeah, you have a peak, um, but the breaking, it's not based solely on the ionic bond status in the protein.
Yeah.
So optimum temperature is governed. So if you think about it, above the optimum temperature, you essentially have breaking of ionic bonds and then breaking of other bonds. If you keep
Yeah.
increasing temperature and you denature the protein, it doesn't work. But if you reduce the temperature of the system, you're not necessarily affecting the rate of reaction by changing the structure as much as you're affecting the rate by changing the kinetic energy of the system.
Okay.
So if you
Okay.
cool
Yeah, makes
cool
sense.
it down, it just reduces
Yeah.
it.
So it's due to like fewer collisions occurring. Yeah.
Yeah, exactly.
Okay.
Okay, question 11.
So it's not polysaccharides.
Yep.
Proteins, yes, and phospholipids, yes. Yeah, 1, 3.
Yeah. Good. That's correct. Next question.
Mm. Okay, hmm. Um, so increasing the temperature by 10, uh, well, it could well, okay, it'll probably like increase the amount of product. So it won't be D.
Why would it increase? Why would increasing?
Well, actually, like we don't really know the optimum temperature of the enzyme.
Yeah, that's a good point.
But like if we're if we if we say that it's probably like an enzyme in our body, then uh it's probably the rate is probably going to increase because 30 Celsius is below our body temperature.
Yeah. Okay, that's a fair point. But also, if you just look at if you look at all of the different initial rates for each of these lines, you can the only options you have are either the same rate or an increased rate. So for line A, you have the same initial rate as line X, but for B, C, and D, you have an increased initial rate of reaction compared to X.
Yeah.
So the choice you have in terms of initial rate change, it either has to stay the same or it has to increase. And
Mm-hmm.
increasing the temperature by 10 degrees, you can pretty much be certain that
It won't be C.
it's not
Okay.
gonna it's not gonna stay the same initial rate.
Yeah.
It's not gonna be A.
Yeah.
Okay.
Well,
Uh,
it's not going to be C either because that's not like enough of a difference.
yeah. Okay. I think what they're trying to show you with C is actually no difference. So yeah, you're
Okay.
right.
So it's probably B.
Yeah, good. It is B. It couldn't be D because you're adding extra substrate, so you would have to therefore increase the amount of product.
But like, if the enzyme, like, it doesn't really state what the optimum temperature is. Like, it could be 25 Celsius.
Yeah, it could be 25 Celsius. That's fair, but if it was 25
Would
Celsius,
it still increase the rate because the substrate increases?
no, it could decrease the rate,
Yeah.
but then you would still have more final product.
Oh, okay. Yeah, because there's more substrate.
Yeah, exactly. The only way this question doesn't make sense is that if in experiment X the the enzyme is completely saturated, and
Yeah,
then in which case adding substrate
wouldn't do anything.
wouldn't increase the amount of product and you would get curve C.
Yeah.
But you can assume, unless they specifically state that, you can assume that adding extra substrate is going to increase the amount of product.
Okay.
Okay, let's move on to 13.
Right. Into the blood plasma. Is it A?
Yeah, it is A. Talk me through your thinking.
The only reason why I said it's A because of the inhibitor. Um, that step would not give you a good estimate for enzyme activity.
Yeah, good. And how about sterilizing blood plasma by heating? Why would you want to not do that?
Well, by heating, you could also like denature the enzyme.
Yeah, you would denature the enzyme if you sterilize plasma.
Yeah.
Um, yeah, so that leaves A. Okay, 14.
Cell produces D.
Yeah, good. Okay.
B.
Yeah.
Oh no!
Sorry.
Wait. Oh no! Yeah, B. Definitely.
Yeah, it's B. So talk me through that.
I just saw recognizing self from non-self, and it's definitely glycoproteins.
Yeah.
The reason why I hesitated is because I just saw proteins too, so I looked if four was present in the first column, but it wasn't. So
Okay. Good. Um, let's go to question 16.
just
So if you just tell me how you would work this out, then I'll let you know if that's correct or not.
okay, yeah. Mm, what shows the ratio? Oh, okay. Oh, okay. So it's where the initial length divided by final length equals 1 because there's no change
Good.
in
And
length. So it would be like about equal to, I guess, C.
yeah, that's correct. Good. Okay, question 17.
Vitamin C. Water. Salt. Okay, so Vitamin D is fat soluble. So that's X.
Good.
So it's either B or C.
Yep.
Then yeah, it's B.
Good. Why is it B?
Because that protein is a channel. While the other one is not a channel or a carrier.
Good. So we have water, the solvent water is essentially moving through Z anyway. So if vitamin C is dissolved in water, it will move through Z. However, if you wanted to move vitamin C through a carrier or an antiporter like Y, then you would have to remove it from water, which would just be inefficient. So you can assume that it's going to be C.
So like it has to be like open in some capacity, otherwise, like how would it go from the environment to the cell, right?
Yeah, it's not. So it is
Or is it?
a transport mechanism,
Mm-hmm.
and that could be. So, for instance, in oxidative phosphorylation in mitochondria, you have ATP synthase, which will sort of move ions into its mechanism, and so Y could function like that in the sense that you could have an ion entering the protein and then moving through the protein and getting into the inside of the cell, but it's not going to be as simple as just a channel which is open, which water can flow through. Does that make sense?
Yeah.
And I think the key for Y as well is that that movement is going to have to be energy driven somehow. So either via facilitated diffusion, where you have movement of ions to sort of generate that movement, or you're going to have heat transport.
Yeah.
Good. Question 18?
No, so it's good. Yeah. Oh, did you say question or did you say question 18?
Yeah, I said question 18. Yeah.
Okay, sorry. Okay. So nuclear division by mitosis doesn't have anything to do with semi-conservative replication of DNA because that occurs in the interphase.
Good.
So it's not B or D. Then question is if it one is. No, it's C
Good. And why is one incorrect?
because the cell divides into two.
Yeah, good. So
Yeah.
the daughter cells are half of the size of the parent cell.
Are they exactly like the same size?
Not necessarily. So in more complex forms of division where you have differentiation of daughter cells and they won't necessarily
Oh
be
yeah, definitely. So like for stem cells, if they divide and then one becomes a differentiated cell and one remains a stem cell, then they might not be the same size.
yeah,
Okay.
but they will be genetically identical.
Yeah.
Okay. Question 19.
Oh. Right. 20.
Okay, so there's 20 in this cell,
20.
and they're saying that this cell has been formed by reductive division.
Reduction. Oh, is this meiosis again? Or
Yeah,
something?
it's meiosis again. So you don't necessarily, I don't think you'll be asked a question like this.
Yeah. But
As we said earlier, in meiosis, it's half, right?
yeah, so it would be 40.
Yeah.
Because gametes fuse together to make like a zygote, which will contain 40. Yeah.
Exactly.
Okay.
Okay.
Mm. Oh my. Okay, right. Okay, so distance between centromeres of sister chromatids is definitely 2.
Good. And why is that?
Because they're next to each other. So the sister chromatids are bound to each other by the centromere up to the anaphase, so it's 0, and then there's an increase.
Yeah. Good.
Um. Okay. Oh. It's so the distance between centromeres and the poles of the spindle is 1.
Good. Explain that to me.
That's because so when the anaphase starts, there's a decrease in that distance because the chromatids are being pulled towards both of the poles.
Yeah, good. So as you have an increase in the distance between the sister chromatids, you're going to have a decrease in the distance between the centromere and the
Yeah.
and the post. Good. And that leaves 3 with the distance between the poles,
Yeah.
which is pretty much unchanged throughout the process.
Okay.
Good.
Um. Oh. Okay. So UAG T A T C A. Wait. A A T A. Oh, it's in between. Okay, sorry. That would be B.
Okay, good. And just quickly talk me through the reasoning behind that.
So DNA. So RNA codon UAG is complementary to ATC triplet code,
Sure.
and so if T is inserted at B, you'll get TC C ATC, and
Yeah.
then
Good. And if I was to remove, let's say, the first base there, if I just took the T out of it completely, would that still be true? Would that mutation still stop the synthesis of a polypeptide?
if you put, sorry, what?
If I just removed the first base of the nucleic acid, if I just rubbed out that first T, so I'm drawing a cross through this T,
Yeah.
and then all you have is the CCA, CP, CG, ATGC. If you introduce that mutation, would you still get a stop codon?
Yeah.
So
Oh no.
you wouldn't actually, because
Why not?
it's read in groups of three, right? So
Oh.
this is
Oh
read,
yeah.
and then this with the mutated T is read,
Okay.
then these are read, etc., etc. So it has to be in sort of groups of three. If you do it one step down from the group of three, then what you get is called a frame shift mutation, and every single amino acid is going to be changed. So every single amino acid coded by that length of DNA is going to be changed to get a frame shift mutation.
Okay, that makes sense. Yeah.
Good.
Okay.
To question 22.
A.
Yep, good.
C.
Good, and why is that?
Because they're three hydrogen bonds, so it's cytosine and guanine, and then guanine is a purine and cytosine is a pyrimidine.
Good.
Okay. I understand. Oh. Oh. Okay. Wait. I might have to write some things down for this one.
That's fine. Yeah, go ahead.
Yeah. C. Yeah. CU. Yeah. Oh, wait. Um. So I know it's not A or it's not. Wait. Yeah, it's not A or it's not C.
Good.
Um. Um. G. Okay. Hmm. Okay. It's B.
It's B or D.
Uh. Maybe not.
So it's actually D,
Oh, okay.
and I'll explain to you how I would go about figuring
Okay.
out.
Because I think I took like a. I think I would have gotten it, but like I did it like a wrong way. Maybe.
Okay, well, let me tell you how I would do it, and then you can let me know if that makes sense to you. So we've got tRNA at the bottom here, and the way I would do it is correspond this tRNA
Oh yeah.
codon to an mRNA code,
Yeah.
and then correspond whether or not that amino acid is
Yeah.
going to be in the sickle cell sequence.
Yeah.
So the first one you have CAU, that corresponds to GUA. You
Mm-hmm.
check whether GUA is here, it is, and
Yeah.
that's part of valine, and then you check if valine is part of sickle cell, and it is.
Mm.
If you
And
do
then,
that with each of these, you'll find that each of them have an amino
yeah,
correspond to
GAA for B, and then yeah, okay. Yeah, I think you know what I did, but I put the mRNA codons into
yeah.
RNA, which took longer. Yeah.
Yeah,
Okay.
if you do it the other way around, you're working with a lot more variables, so you have to write out a lot more, and it takes long.
Yeah, yeah.
That's all. Let's move on then. Um,
D.
uh, okay, why do you say
Well,
D?
I think sucrose is reducing sugar, is it? Or is it a non? No, sucrose is a non-reducing sugar. Okay. Yeah. Then it's A.
Yeah, good thing. And talk me through that. So Benedict's test and iodine and
Yeah. So iodine is so if starch is present, then it turns black, and if it's not, then it remains brown.
good.
So B and C are out, and then when sucrose is hydrolyzed and broken into monosaccharides. Monosaccharides are reducing sugars, so the Benedict's solution would turn from blue to red,
Yeah,
and yeah,
good. Perfect.
just A. Yeah.
Okay, so I don't think you. Let's see. Yeah, okay, you can do this one.
Okay. Um, Oh, we had this. Wait, oh, maybe not. Yeah. It's A.
Yeah, good. So it's exactly the same as the other question, only it's a bit more convoluted. Um, okay, let's move on. So,
A.
why A?
Because so there's no lignified walls that's present in the xylem.
Okay.
And then solute potential of cell content is high because there are a lot of assimilates and sucrose in the sieve tube.
Okay, so you're right, but they're actually asking you about the solute potential of the cell content in the sieve tube, not the solute potential of the content of the sieve tube itself. You understand what I mean?
Okay.
So you've got the tube itself, the space in between the cells. That solute potential is high, but that isn't within the sieve tube cells. If you look at the solute potential of the sieve tube cells, it's actually low because pumping, they're going to be pumping the assimilate into the tube lumen. Does that make sense?
So what they're asking is the solute potential of the companion cells.
Yeah, exactly.
Oh. Okay, but isn't the phloem sieve tube made out of sieve tube elements, which are cells?
Yeah,
But without end walls, but they're still cells.
yeah. So that's sort of what we're talking about here. So those elements, you have the sort of mass flow of water and solutes through them into the tube. But if you looked at the solute potential within the cells, it would be low because all of the water and solute is moving into the tubes themselves. Does that make sense?
Okay.
That's that's the sort of fact that they're trying to. Um, that's the fact that they're trying to test here. You're right. So sieve tube elements are, they consist of sieve tube cells,
Yeah.
um, and basically in those cells you've got the transport of water and solute by diffusion and active transport into the sieve tube itself. And so if you looked at the solute potential in those cells, it would be low compared to the solute potential in the sieve tube.
Okay. Okay, so they're kind of talking about like the like cytoplasm of the like okay. So you would say that the sieve tube has a high solute potential, but the sieve tube elements don't.
Yeah.
But the sieve tube elements make up the sieve tube.
Yeah, but it's so in the sieve tube element, you have pores, which sort of is where the water and the solutes are moving through. Does that make sense?
Okay, that makes sense then. Yeah,
And in the actual cytoplasm of the
there's nothing. Well, there's no sucrose.
yeah, and when they say solute potential of the cell content, they're
They're
plasm.
talking about yeah. Okay.
Okay. Yeah, sorry, a bit of a convoluted explanation there,
No,
but
that's good.
let's move on. So 28.
Well, okay, so the okay, the heart beats once every 0.75 seconds.
Good.
And then I would just have to calculate how many it would do in 60 seconds.
And what's the calculation?
So 60 divided by 0.75,
Good.
which is 75. Yeah.
It's 80, but don't worry.
Or 80 or whatever. Okay.
We'll read off the calculator.
Yeah.
Okay. Question 29.
Oh. By cost. Oh. Left. Okay, wait. So semilunar valve opens at 2, closes. Okay, so it's not B or A.
Talk me through why you think that is.
Huh?
Why do you think that is?
Because so the semilunar valve is between the left ventricle and the aorta. And so at 2 is the moment where the pressure of the blood in the aorta is bigger than the pressure in the left ventricle. So, it's closed in 2. Okay. Yep. So it's not COD.
Yeah, that's right.
And then, okay, for the left atrium, when it's bigger, it opens. So it's B.
Good. Now talk me through that bicuspid valve part.
Okay. So when the pressure of the left atrium succeeds the pressure of the left ventricle, the bicuspid valve opens and the blood flows from the left atrium to the left ventricle.
Yeah.
So, which is at 4.
I mean, you could also think of it in the sense that the bicuspid valve closes when the pressure in the ventricle begins to rise.
Yeah.
Good. Maybe.
Okay, so rolled leaves. Oh, okay, wait. Sorry. Leaves. Swollen leaves. Increase the distance. A.
Why A?
Because rolled leaves decrease water loss by transpiration. Because it remains humid, the outside of the stomata.
Yeah, good.
And then swollen leaves will increase the distance for transpiration to actually wait. Could it be D?
Yeah.
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 Okay. Because I thought that swollen leaves would increase the distance for transpiration to occur, but it doesn't really, because transpiration occurs through the stomata. So it would make some cells more further away from the environment, but it wouldn't decrease transpiration rate.
Yeah. So transpiration is largely influenced by the interface between the stomata and the external environment. So for all of the other options other than A, you're changing the environment that the stomata is in. But sorry, in all the conditions apart from swollen leaves. But with swollen leaves, you're not really changing the fact that the stomata are, exposed to the element, and therefore you're not really changing the rate of transpiration.
Yeah.
Okay. 31.
D.
Okay, good. And explain that to me.
Because one is definitely correct. All other men are more at risk of dying from lung cancer than male doctors. Because like the line for all other men is above that of male doctors.
Yeah.
So it's not B or C, so it's A or D. Then I checked whether or not 3 or 4 was correct. And so male doctors gave up uh, smoking cigarettes than all other men because there's a decrease in the line. Like, yeah.
Good. Yeah. That's it. 32.
Nicotine.
Good. 33.
Bonkers. A.
Yep, that's correct. 34.
Okay. It's not A.
Good.
It's not B.
Yep.
It's D.
Okay. Why isn't it C?
Because I think D is correct.
Okay. So if D wasn't there, why would you say that C is incorrect?
If what was there?
So if D wasn't an option, why would you say C is incorrect?
Um. So I don't know what atheroma buildup is, but I guess it would be something like fat buildup in the inner of the arteries.
That's correct.
Okay. Um, and that wouldn't improve. Oh no. Well, it could be C, but I don't know. But I thought D was just correct.
Okay.
Okay.
So the key here is that they're asking you how would the health improve of the person suffering from mild emphysema. So E and D are technically both correct in that both of them would incur, both of these options would occur if you stopped smoking, but one of them is going to improve the health of the person, whilst the other one isn't necessarily going to impact on their health.
Okay.
So the answer is C. Sorry, the answer is D. But so you were right. But um, the fact is, with less atheroma buildup on the inner lining of the arteries, you're not necessarily going to increase the lumen diameter because it's already decreased from the buildup. Does that make sense?
Sorry again. I didn't really get that.
That's okay. So with less atheroma buildup on the inner lining of the arteries, you're not going to increase the lumen diameter. It's just going to stay the same.
Oh yeah, definitely. Yeah. The diameter anyway stays the same. Okay. That
So
makes
that's why you eliminate C. So it is D. You're correct in the first one.
okay.
Okay, question 35.
Sorry, you said sorry, back to 34. Sorry, you said B would be correct. Like that would happen if you stop smoking cigarettes.
Yeah, that is correct.
Um, why?
So when you stop smoking cigarettes, you decrease the carbon monoxide level in the blood.
Yeah.
And so as carbon monoxide is gradually removed from the blood, you have less carboxyhemoglobin being produced.
Yeah.
And so instead, hemoglobin becomes occupied by oxygen. Therefore,
Okay.
the oxygen carrying capacity of the person increases.
Okay, but how would the oxygen capacity affect the number of phagocytic macrophages arriving in the lungs? That's
Sorry, no.
I'm talking about B.
No, yeah, no. B is wrong.
Yeah. Ah, okay. I thought you said B was correct.
No. The answer is D. The answer
Okay,
is
then it's over. Yeah.
no problem. 35.
Um. Which disease is treated with drugs that have a similar structure to DNA nucleotides? Um. So it's not cholera because cholera is treated by oil rehydration.
Good.
The other three are treated with drugs, or can be treated with drugs.
What kind of drugs are the other three?
So I know that, well, anti-malarial drugs or prophylactic drugs.
Yeah.
Tuberculosis. I know that they can be treated by another type of drug. So I don't know exactly which type of drug there. I don't know if we have to know that, or maybe we do, but I've never.
That's fine. So I think the key here is kind of working out by elimination. So as you say, cholera is treated with rehydration therapy. So that's not in. Malaria and tuberculosis uh, are both in. So HIV, malaria, and tuberculosis are all caused by pathogens.
Yeah.
HIV is virus, malaria is parasite, and tuberculosis is bacteria.
Mm-hmm.
So malaria and tuberculosis are going to be treated by anti-parasitic drugs and antibiotic drugs or antibacterial drugs.
Mm-hmm.
And HIV/AIDS is treated with a variety of antiretroviral drugs.
Mm-hmm.
And, you know, I'll double check this. I'll double check to see if you need to know like specifically that the HIV treatment has a similar molecular structure to nucleotides.
Okay.
But that's the answer. It's HIV/AIDS, and you can maybe work out. I guess it is a bit tenuous, but you can maybe work it out from the sense that a antibiotic that stops a bacteria or a parasite is going to have to do more than just replicate the nucleotide sequence, because the way that the HIV antiretroviral therapies work is that the nucleotide or the molecules that are analogous to nucleotides basically disrupt the enzyme action of HIV and AIDS.
Yeah.
HIV enzymes use these DNA nucleotides instead of the actual HIV RNA, and that's what sort of suppresses the virus in the body. But yeah, I'll double check that. But okay, we're done. That's us done for today. Next week, do you want to go through same kind of stuff, paper ones, or would you like to do a mix, or what would you like?
I think we should stick with paper ones. That would be good.
Okay, sure. All right.
Okay. Thank you.
All right. Thanks, Marcos. Speak to you next week.
See you. Bye.